Imagine a war so small you can't see it, yet it shapes the course of your life every single day. Invisible invaders, viruses, bacteria, toxins, slip into your body, seeking to exploit your cells. On the other side, an equally invisible army stands ready, the immune system, a vast network of cells and molecules trained to detect, respond, and remember. At the center of this microscopic conflict are two central players, the antigens that mark the enemy, and the antibodies, also called immunoglobulins, that neutralize them with astonishing precision. Understanding how this battle works is the foundation of microbiology and immunology, and the deeper we explore, the more ingenious and intricate it becomes. An antigen is any molecule capable of triggering an immune response. Most often it comes from a pathogen, a protein on a viral capsid, a sugar on a bacterial cell wall, or a toxin released into the bloodstream. These antigens are recognized because they look foreign compared to the molecules normally found in our own bodies. On their surfaces, antigens present unique features called epitopes, small regions where antibodies can latch on like a lock meeting its matching key. The key itself, our antibodies, are large, Y-shaped proteins produced by B lymphocytes. Each arm of the Y contains a variable region, a sequence of amino acids precisely folded to bind to just one type of epitope. This specificity is so fine-tuned that an antibody raised against one strain of influenza may not recognize another strain with even a single amino acid change in its surface proteins. The range of antibodies, known as immunoglobulins, comes in several classes: IgG, IgA, IgM, IgA, and IgD, each adapted to a different kind of mission. IgG is the most common in the blood, versatile and long-lasting, capable of crossing the placenta to protect newborns. IgA patrols mucosal surfaces in the gut and respiratory tract. IgM is the first responder, forming large pentameric complexes that rapidly agglutinate invaders. IgA triggers allergic responses but also plays a role in defending against parasitic worms. IgD, though less understood, seems to help activate B cells. These molecules do not act in isolation, they are supported by the rest of the immune system, especially the cellular branch that provides backup when antibody responses are not enough. But here's a mystery: the human genome has only about 20,000 genes, yet our bodies can produce millions of distinct antibodies. This is possible because antibody diversity is not pre-written in our DNA as separate genes. Instead, it is generated through a remarkable genetic shuffling process. In immature B cells, the genes encoding the antibody's variable region are assembled from separate segments: V, variable, D, diversity, and J, joining, segments for the heavy chain, and V and J segments for the light chain. Through a random recombination process, mediated by enzymes like RAG1 and RAG2, different segments are stitched together in countless combinations. Even more diversity arises from the random addition or deletion of nucleotides at the joining sites. And later, from somatic hypermutation, point mutations introduced into the variable region during B cell activation in lymphoid tissues. This molecular lottery ensures that before you are even exposed to an antigen, your immune system already has B cells capable of recognizing an almost infinite range of possible invaders. The moment a pathogen breaches your physical barriers, the immune system's two main branches, humoral and cell-mediated, swing into action. While antibodies dominate the humoral response, the cell-mediated arm is run by T lymphocytes. Cytotoxic T cells directly kill infected host cells, especially critical when dealing with viruses that hide inside cellular machinery. Helper T cells coordinate the attack, releasing cytokines that activate B cells, macrophages, and other immune cells. This interplay ensures that the body doesn't just recognize a foreign antigen floating in the bloodstream but can also hunt down and destroy cells where the antigen is hidden away. When an antigen is first detected, antigen-presenting cells such as dendritic cells capture and process it, displaying its epitopes on their surface bound to MHC molecules. This display is like a wanted poster shown to passing T cells in the lymph nodes. Activated helper T cells then provide the essential second signal to B cells that have bound the same antigen via their surface immunoglobulin. The B cell undergoes clonal expansion, rapid cell division producing many identical copies, and differentiation into plasma cells, which are antibody-secreting factories. Over the course of days, these plasma cells can churn out thousands of antibodies per second. Meanwhile, some B cells become memory cells, lying in wait so that if the same antigen is encountered again, the immune system can mount a faster and stronger secondary response. Viruses, however, are not passive targets. Their replication strategies in microbiology are a masterclass in efficiency. A typical viral replication cycle begins with attachment, specific viral proteins binding to host cell receptors. Then comes penetration, either by fusion with the cell membrane, endocytosis, or, in the case of bacteriophages, direct injection of genetic material. Once inside, the virus uncoats, releasing its genome into the host cell. The method of genome replication depends on the type of virus. DNA viruses often head for the nucleus to use host DNA polymerases, while RNA viruses typically stay in the cytoplasm, bringing their own RNA-dependent RNA polymerases. Retroviruses like HIV reverse transcribe their RNA into DNA, integrating it into the host genome so that the cell itself becomes a viral factory. After replication and synthesis of structural proteins, viral components assemble into new virions, which exit by budding or lysis, ready to infect new cells. The morphology of viruses varies greatly, influencing how they attach, enter, and evade the immune system. Some, like adenoviruses, are non-enveloped with icosahedral symmetry, making them robust against environmental conditions. Others, like influenza or HIV, are enveloped, with lipid bilayers stolen from host membranes and studded with glycoproteins essential for entry. Still others, such as bacteriophages, have complex morphologies with icosahedral heads and helical tails, specialized for injecting nucleic acids into bacteria. These shapes are not just visual curiosities, they reflect evolutionary adaptations to host range, stability, and immune evasion. Understanding viral morphology and replication is crucial because it shapes how the immune system recognizes and responds to infection. For example, antibodies may neutralize a virus by binding to surface proteins, blocking receptor attachment. This is the principle behind many vaccines. By exposing the immune system to viral antigens in a harmless form, we train it to produce neutralizing antibodies before the real pathogen arrives. In natural infections, neutralizing antibodies can prevent viruses from infecting new cells, while opsonizing antibodies tag viral particles for destruction by phagocytes. But the virus-immune system battle is dynamic. Viruses mutate, influenza's hemagglutinin and neuraminidase proteins change regularly, requiring updated vaccines. HIV's high mutation rate creates a swarm of quasi-species inside a single individual, making it hard for antibodies to keep pace. Some viruses hide their antigens inside host membranes or downregulate MHC molecules to avoid T-cell recognition. In response, the immune system leans more heavily on cell-mediated defenses, with cytotoxic T lymphocytes scanning cells for abnormal peptide presentation and triggering apoptosis in infected targets. The molecular basis of antibody production links microbiology, immunology, and biochemistry in one seamless process. Once a B cell is activated, its antibody genes are transcribed into mRNA, translated into polypeptide chains, and assembled in the rough endoplasmic reticulum. Heavy and light chains are folded and joined by disulfide bonds, with glycosylation occurring in the Golgi apparatus to stabilize structure and function. The final antibody is secreted via exocytosis or expressed on the B cell surface. Class switching, changing the constant region of the heavy chain without altering the variable region, allows the same antigen specificity to be delivered in different functional forms, such as switching from IgM to IgG during the course of an infection. In the bigger picture, the antigen-antibody reaction is the visible tip of a much deeper iceberg of immune complexity. Antibodies bind antigens via a combination of hydrogen bonds, hydrophobic interactions, and electrostatic forces, non-covalent yet highly specific. The strength of this binding, the affinity, is refined over time through affinity maturation, as B cells producing higher affinity antibodies are selectively expanded in germinal centers. This continuous improvement means the immune response becomes more effective as an infection progresses. In the lab, microbiologists use antigen-antibody reactions not just to study immunity but to diagnose disease. Agglutination tests detect particulate antigens like bacterial cells. Precipitation tests detect soluble antigens. ELISA assays can measure either antigens or antibodies with high sensitivity. Western blots can confirm the presence of specific pathogen proteins. These tools are built on the same fundamental specificity that protects us in vivo. By harnessing the immune system's precision, we can detect infections earlier, track outbreaks, and design more effective vaccines. As the fight between viruses and immune defenses plays out in every living organism, we are constantly reminded that this is an evolutionary arms race. Viruses refine their replication and evasion strategies, the immune system evolves mechanisms to detect and neutralize them. The diversity of antibodies, the precision of cell-mediated responses, the structural ingenuity of viruses, and the complex biochemical pathways that produce these molecular defenders all form an interconnected web of survival. This is not just a battle for the present infection, it is a rehearsal for the future, embedding memory so that the next encounter will be met with a faster, stronger response. In this hidden war, victory is measured not in destroyed enemies, but in restored health. Each time you recover from an infection, it is the result of millions of perfectly matched antigen-antibody interactions, coordinated with cellular strikes against infected hosts, and the biochemical machinery that makes it all possible. Microbiology shows us the enemy's playbook, immunology and biochemistry reveal our defenses, and together they tell the story of how life itself resists the forces that try to end it. This story is ongoing, and in every one of us, the next chapter is already being written.